So for our for our Limnoseries today, we are welcoming Anderson Dominguez as our speaker. Anderson is a biologist and a master student at the National University of San Marcos in Lima, Peru. And he's working on taxonomy and ecology of microalgae and cyanobacteria. Before that, Anderson studied his bachelor program in biological science at the same university in San Marcos. And he is now a member of the Department of Limnology of the Natural History Museum of his university and specialized in management and environmental assessment impact. Now, I would like to give you the floor, Anderson. We are all very excited to hear about your talk. So I'll let you share your screen now. The floor is yours.
Hi, can you hear me well? Yeah, thank you. Okay, I'm going to share my. Mhm. Okay, so, wait. Thank you, Lisa. Thank you, Glenn, for the opportunity to talk about some of the research in my lab. I'm very excited to present it for Gleo because I consider it's a good organization which includes many students and scientists from many countries. And also thanks to the attendance because I am not at a time difference. So here we go. I'm going to talk about microalgae and bacteria in tropical aquatic ecosystems from Peru. And I will focus on cases and challenges because it's important to study these biological communities and the potential effects on diversity and human beings. So, and phytoplankton is a crucial component in aquatic food webs and plays a crucial role in determining the structure function of aquatic ecosystems. Understanding also the process and mechanism and how phytoplankton community responds to environmental changes is critical to for the community ecology and also of great significance for aquatic ecosystem management. And also in aquatic ecosystems, it has become increasingly clear that temporal duration in abundance and composition of phytoplankton, which includes microalgae and cyanobacteria, depends on the change of environmental conditions. And these variations of the environmental parameters can also affect in phytoplankton in different ways. For example, in the species diversity or functional diversity, based, for example, on traits, they could be affected, or also for another trophic levels, for example, so plankton, macroinvertebrates, fish in another communities. And also phytoplankton are primarily controlled by temperature, light, water fluctuation, and also predation intensity, which could cause some events, for example, freshwater cyanobacteria blooms. These blooms are a major threat to freshwater ecosystem, and it's strongly associated with shifts in phytoplankton community structure. And in freshwater cyanobacteria blooms, also that is the uncontrolled increase for one or more populations that may can be present for days, weeks, even months. So that means that the cause of algae or cyanobacteria blooms, sorry, that means that cyanobacteria bloom event is eutrophication, which is the altered increase of nutrients such as nitrate, phosphate from wastewater, agriculture, rising temperatures, also related with the climate change. For example, at high levels of temperature can affect the phytoplankton community. According also with the freshwater cyanobacteria blooms, with the extension and intensification of cyanobacteria blooms in freshwater ecosystems, the ecological effects have aroused wide attention from many scientists. Also, the frequency, intensity, and duration of cyanobacteria blooms are increasing worldwide. Also, it's impacting economy, ecosystem services, for example, and also for human health, and leading to the decline of that diversity and also the destruction of ecosystem processes. Also, the process of bloom formation has a destructive effect on phytoplankton community structure because of the dominance of certain phytoplankton species. These cyanobacteria blooms are believed to be a threat that cause increased biodiversity in water ecosystems due to the numerous inhibitory effects on the other communities, like, for example, zooplankton, which for the toxin that production, or for nutritional values or morphology. So there is also many different kinds of bloom. For example, they could be done by cyanobacteria, Limnoseries, chlorophyta, or could be a mix. Algae blooms in general can form by different taxa, including these groups, and they can generate different changes in the characteristics of the water, or also can affect other populations, includes other microalgae and cyanobacteria and zooplankton. But cyanobacteria, according to this article done by Amorin and Mundo, they mentioned that cyanobacteria and mixed blooms intensify the deterioration of water quality, which have become more turbid, acidified, saline, alkaline, and deeper eutrophic. Also, in cyanobacteria blooms, the most harmful consequence is related to cyanotoxin production, which is a serious and important impact for aquatic ecosystems. The increase of the turbidity caused by cyanobacteria blooms has also a serious consequence on pelagic and benthic communities because it reduces the prey visual capture and forcing animals to migrate to another sites, which there is more where there's a more probability to get a prey, for example. So in the case of Limnoseries or Limnoseries blooms, they create a cascading effect on zooplankton because they increase the biomass of large herbivorous station, for example, and consequently decreasing the phytoplankton richness. And some cyanobacterial species have uh, the potential to produce compounds and toxicity, which is estimated to occur in a majority of all cyanobacteria blooms in worldwide. The cyanotoxins are secondary compounds or metabolites that produced by some species of cyanobacteria, and they have potential biological activity, and they can cause effects even in humans, also in terrestrial animals, in aquatic organisms, and environment, obviously. So the most common and better studied cyanotoxins are microalgae, nodularin, anatoxin, platitoxin, and cylindropermol. Based on their toxicological target, cyanotoxins can be classified also as hepatotoxins, cytotoxins, neurotoxins, and dermatoxins. But also microtoxins are the most prevalent toxins in the fresh water and are also among the potential cyanotoxins. And these cyanotoxins, especially hepatotoxins, may affect the health of humans or other animals caused by contaminated where they drink the water, for example. And according to World Health Organization, they establish reference limit values for cyanotoxins in drinking water and recreational waters as well. But how to detect and quantify a bloom? In order to alert to or predict the impact of blooms, a comprehensive report is required to establish that includes the multiple ecological and toxicological aspects in each environment, will be a lagoon, reservoir, or a wetland. So each environment have to be include a typical method or technical according to the human resource condition as well. For example, we can use visual monitoring, satellite image, 5 fluorometric probe, or spectrophotometer. But I'm going to talk of each of these methods. For example, some indirect methods that provide us with only a warming, easy visualization of water bodies that present brown or green colors. So these characteristics could alert us early on about the possible event of bloom. Another method is the use of satellite image, which is based on the concentration peaking measurement. So the most common has been also a spectrophotometer or fluorometric quantification, or which measure the chlorophyll A in organic tracts. But in a prior observation of the sample, have to be carried out to corroborate the dominance of species of cyanobacteria. More current alternatives include also fluorescence and of chlorophyll A and marker pigments of cyanobacteria. For example, glycosyanin, which are measured with submersible probes or handheld fluorometers like this. And the use of remote sensors is uh, also being used as a monitoring tool for living water bodies, which is affected by cyanobacteria blooms. It through the application of algorithms to estimate the concentration of chlorophyll A or phycocyanin from the analysis of satellite image. Now, I'm going to talk about some tropical aquatic ecosystems in Peru. In Peru, there are various aquatic ecosystems because of its geography that can be classified on coastal, high Andean, or Amazon zones. So along the coast, aquatic ecosystems are wetlands, which are considered resting areas for migratory birds, and they are considered to have a great productivity in these ecosystems, according to many studies, because this ecosystem, there is a big diversity of phytoplankton, zooplankton, macroalgae, which is the food for many birds. Other ecosystems are lakes, lagoons, rivers, and high Andean wetlands. They provide goods and services such as drinking water and recreational areas. Rivers are also present in the Amazon, like where they are important because of the great diversity. Among the aquatic ecosystems, the most recognized is Lake Titicaca, which is shared by Peru and Bolivia. It's considered like the second largest lake in South America and the highest navigable lake in the world. Also, this lake is divided into three areas: Puno Bay, Greater Lake, and smaller lake. About their ecosystem goods and services, well, this lake provides different ecosystem goods and services. Among the activities carried out is artisanal fishing that extracts fish, for example, orestias and Titicaca, which in Spanish we say truchas. And also, this provides another activity that in recent years, agriculture has also increased, which has generated economic income for the local population. This has, however, brought negative impact for biological diversity because the high production of fish and the introduction also of another species, for example, odontestes and oncorhynchus, can alter the food web in this kind of ecosystem. And obviously, the increase in nutrients in the water from the aquaculture waste. And also another important activity is national and international tourism, because the reconnection of the lake, about also the culture, the particular characteristics such as the floating houses, the float on the water, which were built by Stora, which are aquatic plankton. and also the presence of this aquatic plant uh, is used for the elaboration of crabs, houses, and boats. However, sadly, all these activities have generated negative impacts on the lake. There is particularly no water treatment plant now. So residential and commercial waste is dumped directly in the lake. So it increased the levels of nutrients and contamination in the water. Also, this has caused acceleration of the eutrophication process, like we can see in these pictures, mainly in the inner bay of Puno, which is the site where there are more tourism and human activities. So for that reason, in the lab, we present a project of DNA metabarcoding and morphological identification of microalgae and cyanobacteria focused on biological indicators to assess the water quality of Lake Titicaca. We this project from 2019, and I will present some preliminary data of for only morphological microalgae and bacteria of this project. And we determined here 19 sites of monitoring around Peruvian area, only for Peru. But now I will show you some interesting data of Puno Bay. This bay is divided in 3 zones. Yeah, the first is the inner bay near the city of Puno. It's the most touristic. The another zone is the outer bay of Puno. And between both zones, there is a large extension of aquatic plants that looks like a barrier in these parts. So these zones are connected by 2 channels, one located in the north, and another in the south, which we call in the project the channel transition zones. So here are located also the floating the floating houses made of aquatic plants, or in Spanish, flora. So in this bay, we determined 9 monitoring points where we took physicochemical data and qualitative and quantitative phytoplankton. So according to some results, Puno Bay presents typical physicochemical characteristics of high Andean lake. And most of the physicochemical variables affected the permissible limits in water according to the environmental quality standards in Peru, such as, for example, conductivity, the total dissolved solid, and nitrate, and total phosphorus that are in the example. So the highest concentration of nitrate, for example, were during the wet season. It's probably due to the entry from external areas of nutrients such as nitrogen and phosphorus that can increase with rainfall. Also, the highest values of nutrients were in the inner bay, which is located near to Puno City. and also in the transition zones through canal, because the population threw up the wastewater directly to the lake, specifically to this site. Likewise, the increase in nutrients could be caused by another anthropic activities, for example, agriculture or livestock, which are a common activities around the Titicaca Lake. And also another, according to another result, but in species richness, a total of 85 taxa of microalgae and cyanobacteria were identified between species and taxonomic forms during the research period. And among the diatoms, which heterocontophita, the most frequent were Flagellaria crotonensis and Albufera granulated. Of the chlorophytes, the most representative were Pseudophyllus boreanum and Phocistis. Among the cyanobacteria were more represented by Snowella, Ephinocapta, Compostvaria, and Microcystis. In addition, an interesting report was Serration hirundinella, since it's considered an invasive species that I'm going to talk a little more in the next slide. This richness will be also because of the diversity of habitats that the lake represents, such as aquatic plants, for example, flora, and favorable phytochemical parameters or conditions for the development of some specifically species. According to the abundance of phytoplankton, the species which were more highest in abundance were Albufera, Osis, Leucosphaera, granulate, Compostvaria, and Snowella, and Microcystis aeruginosa. Among a total abundance of phytoplankton, the inner bay of Puno has the highest abundance with a percentage of 81.87%, followed by the transition of channels with 10.61% of the abundance. And the outer bay of Puno with that 7.52% of the total abundance. And why there is a difference between the abundance in these zones, all these by Bay of Puno? Well, this wide difference in the abundance between these three areas could be because of the morphometry characteristics of the inner bay and the presence of aquatic vegetation for aquatic plants, in this case, Totora, that could limit the circulation of water. Therefore, it will not allow the rapid flow of phytoplankton nutrients or some nutrients to the outer bay of the lake. As a result of this project, we have published a publication on invasive species of Serration, which provides information of the system in Peru of two invasive species of Serration, which is Serration Hirundinella and Serratium porcoides. This study was we take data from many and different lakes and reservoirs around Peru, and the occurrence of Serratium hirundinella and Serratium porcoides has generated problems in the purification process of water sources, causing changes in the organoleptic properties, the formation of non-toxic algae blooms, and some causes in fish mortality, for example, due to the hypoxia of in these water bodies. Another aquatic ecosystem in Peru is coastline wetland. These aquatic ecosystems are characterized by the mixing of fresh and salt water, but they have a rich in nutrients. And this characteristic is the responsible for different gradients that determine the availability of light. Also, another parameter is the nutrient and salinity for the biota in general. So the change in this chemical and physical gradient greatly influence in the abundance, in the diversity, in the spatial temporal distribution and functional characteristics of biological communities. Also, this wetland provides essential ecosystem services, for example, flood mitigation, coastal erosion, hydrological and ecological processes, and recreational activities. However, in recent years, some threats have increased, such as habitat alteration, invasive species, and decrease of production of solid and liquid waste. These changes in water bodies influence also in the individual, population, community, and ecosystem response of organisms, including microalgae, cyanobacteria that cause change in the diversity and abundance of another species. That is why we are also carrying out a project which is the potential toxic algae bloom in coastal wetlands from central Peru. We study microalgae and cyanobacteria of two wetlands in Lima. The first is Albufera the Medio Mundo, which is characterized as a regional conservation area by the Peruvian state. And we were interested in this place because we observed many dead fish and proliferation in the algae, which is Java. So it was interesting for us because it also allowed us to ask what is happening in this wetland. Another wetland is El Paraíso, which has no conservation status, and it's made of two sectors, a North Lagoon and a South Lagoon. In the North Lagoon, like you can see in this video, we were also able to observe the fish, and also in this area, there is artisanal fishing and tutora extraction and carried out while the South Lagoon is an area and used to recreational purposes. However, another, this is the video of another side, the southern sector, where there is no additional fishing activity, but it's an area which people use like a recreational. So I'm going to also present some preliminary data according to the physicochemical parameters. The characteristics were consistent with the coastal environment, where we can see high temperatures, high salinity levels, and alkalinity waters. However, the levels of nutrients like nitrates and phosphorus were higher than the standard in the Peruvian states. And in another wetland, in Paraíso, the physicochemical characteristics were typically of coastal ecosystems. In addition to be less deep, this lagoon show slight differences with the North Lagoon and also lower than standard values for the nitrates and total phosphorus. So this is probably due to the fact that in the north area of El Paraíso, there are wastewater effluents and less water flow since there's a greater extension of Soranid. And according to the species richness, a total of 36 sets of microalgae or cyanobacteria were recorded, distributed in 6 phyla, 6 classes, 21 orders, and 27 families. In both aquatic environments, cyanobacteria such as Microcystis protocystis, Microcystis brasiliensis, and Lemermaniella and Colomoron were predominant in these two wetlands. Also, some diatoms, such as Glaciotropis and the Actophyta, which is considered an organism that generates toxins during blooms events. Also, there are some reports of cyanobacteria and cyanotoksin in Peru. Several studies was carried out, but they are still insufficient. For example, Markova, Montoya, and Komarek confirm the presence of important planktonic species, which is potentially causing heavy water blooms in Puno Bay. And also, the most frequent planktonic cyanobacteria was Limnoseries robusta. However, it's not danger of blooms of Limnoseries robusta appearing in this bay, since there is no niche for it, for this species. Another study was carried out by Leonardo Mendoza, who recorded in Huacachina Lagoon the species Spirocarbon brasiliensis and Microcystis westernbergi, which now they are both in the Microcystis genus. Also, another report was carried out by in cyanotoksin. But the first report in cyanotoksin, it's a little interesting because the first report was of hepatotoxin microsystem in a recreational lagoon, but it was published in an abstract that was part of an academic event. However, in this year, a new report of microsystems cylindropermoptin, tatiopsin, and anatopsin in arterial was published. So it could show us that we need to study the cyanobacteria in many reservoir lagoons, lake, because we don't know what is the concentration of the cyanobacteria in these aquatic ecosystems and how they can affect to another trophic levels. Okay. So according to monitoring and monitoring regarding bacteria in Peru, there is a scientist who is Anthony Salazar, who is constantly adding the bacteria and cyanobacteria in Peru. Also, there is a national water authority that monitor different reservoir and lakes. However, some parameters like phytoplankton and cyanobacteria is not be considered on this monitoring activity. And also, like this scientist Salazar is investigating, or it's part of the Autoridad Autónoma de Market, which is an institution that includes a monitoring program that incorporates identification and quantification of water based on microscopy. So they are using a framework to develop a monitoring plan of the drinking water. And also, it's really important to for the management and conservation. So this framework includes 3 levels of awareness. The first is level 1 is the vigilance level, where the abundance of cells will be 200 cells per millimeter of cyanobacteria. The second is alert level 2, where it will be 2,000 cells per millimeter of cyanobacteria. And the third is alert level 2, where there is 100,000 cells for cyanobacteria. But also, there is a published of Peru and welfare based on cyanobacteria biovolume. So they also classify this level in vigilance level, alert level 1, and alert level 2. Besides of many reports and the work that many institutions make, there is a regulatory framework regarding cyanobacteria and cyanotoxins in Peru. Peru has 5 core regulations regarding the evaluation of water quality. The first is national strategy to improve the quality of water resources. And another is the national protocol for the monitoring of the quality of water resources. There is the classification of the surface continental water bodies, the environmental quality standard for water, and the regulation of water quality for human consumption. Also, the Peruvian states give quality standard for water, and they classify in 4 categories. The first, population and recreational. It includes reservoirs, lakes and wetlands that people use for make tourism, for example. The second category is coastal and island marine extraction and farming activities. The third is irrigation of vegetables and animal drinking. And the fourth is the conservation of the aquatic environment. And also, there is a value of cyanotoxins that this quality standard for water mentions that the limit is 1.0 micrograms per liter of this cyanotoxin in the water, drinking water. So all these state of the study of cyanotoxins and cyanobacteria it have some challenges. The first is monitoring plants in lakes, lagos, and reservoirs in local periods, which allows us to a better understanding how is the development of this community of phytoplankton. Second challenge is implementation of the early warning alert of blooms. The third is implementation of methodologies for quantification of plankton abandon. The fourth is the implementation of thresholds for cyanobacteria abandon alert levels in drinking water in recreational areas. And the last, investigation of potential drivers of contamination, because it's important to identify what is the cause or the principal cause which generates these events, and also as a consequence, they have a negative impact on the another biological communities and also for humans. So thank you for your attention, and I am very excited with some for any question.